thank you to the organizer for the opportunity to talk here at this important event. So I'm going to talk about the immune response in Chagas disease, which is a challenging mission and to do so in a short time, but I'll try to give you a general view of it. And I will focus on the T-cell responses, which we've been studying for the last years at my laboratory at the Federal University of Rio de Janeiro. And particularly, we study cytotoxic CD4 cells, which may sound a little bit strange, but I will go through it. Okay, so, but before, I would like to remember the importance of basic research. We would not have had the vaccine against COVID so quickly if without all the years of basic research previous to the pandemic. So basic research is important for vaccine development, for treatment development, and for any kind of innovation. Okay, so here is the, I won't go in details in the Trypanosoma cruzi life cycle. I'll just point some important points. After the trypanosoma, the triple mastigote form enters the bite wound site, it will infect different kind of cells, macrophages, fibroblasts, muscle fibers, and it will transform into a mastigote form, which is intracellular. So it grows inside the cells. And after 3 to 5 days, it is released. And then it will, in fact, it is released as a triple mastigote form again, and it will infect the nearby cells that are nearby the one that was infected before, but it also can go to the bloodstream. It goes to the bloodstream, and the bloodstream will bring the parasites to almost every organ, and then it could infect many other cell types. And then it goes on over and over again until the immune response keep it in check. And while in the blood, of course, it can be picked up by another insect and then complete the life cycle. Okay, so the disease has two phases. The acute phase is the one where we have high levels of parasite in the blood and in the tissues, and we have an intense activation of the immune system during this phase that can last for weeks, up to months. But then the immune response will control the parasites. I mean, the levels of the parasites in tissues and blood go down, but is never completely eliminated. So, however, it's important to note that 5% of the infected individuals die in the acute phase. Okay, but then most of persons we enter the chronic phase when the parasite levels are low, as I just said, and most of the patients will stay asymptomatic for the lifetime. However, most of them do have histopathological alterations, and around 30%, sorry, around 30% of the patients will develop the symptomatic chronic phase of the disease. And here we have the big heart and the mega colon, because we can have cardiac form, but also digestive forms is also happens in these patients that develop the symptomatic chronic phase. And this phase is characterized by parasite elicited inflammation and pathogenesis, but there is also an autoimmune component, although there is a lot of discussion about the role of autoimmunity. It's still an important issue to be studied. Okay, and autoimmunity occurs because tolerance to self is broken, so you're not tolerant to your own antigens, components anymore. But the thing is that not only in Cruzi, but following many types of infection, we can have autoimmunity because pathogens can mimicry the self components of the host. So we have here a list of antigens from Trypanosoma cruzi, which are recognized by our antibodies and T cells, but antibodies and T cells cross-react, recognizing self components. So for example, I put here the heavy chain of the human cardiac myosin is as an example of a self-antigen that can be the target of T cells that were activated in response to Cruzi antigens. Okay, so let's have a more general. Of course, I don't have time to go in details, but the first thing that happens when the Cruzi enters the host is the activation of the innate response. Is the first response we have, and so the cells of the innate response, like macrophages, neutrophils, dendritic cells, they got activated because they recognize pathogen-associated molecular patterns that are recognized by receptor of a family of receptors of the innate immunity. And this will trigger the release of several soluble factors, cytokines, and also you also induce microbicidal molecules, which can kill the parasite. But this is not enough. In fact, we also need the response of the adaptive or acquired response, which is mediated by T and B lymphocytes. And sorry. So persons that are immunosuppressed or immunocompromised, they would have much higher parasite levels and can even die. So the acquired immunity is really necessary to control the parasite levels. But as I said, even the acquired immunity never succeeds to completely eliminate the parasites, which go, so we have a chronic infection that lasts for life. And just remind you, so since it was a broad audience, I just want to remind you a little bit about lymphocytes. So lymphocytes, the B cells are the ones that produce antibodies, and the T cells, we have two main subsets of T cells. The CD8 cells are the ones that recognize the infected cells, and they kill the infected cells. So they are called cytotoxic cells or CTLs, while CD40 cells produce large amounts of cytokines, which are soluble factors that act on other kinds of cells, inducing different events. And these cells are called helper T cells, okay, or TH. Okay, so gamma interferon, which is produced by this special type of CD4 cells, the TH1, gamma interferon will allow the B cells to change the kind of antibody they produce. So they will produce antibodies that are important for controlling the parasite too. And the gamma interferon will help macrophages to enhance the production of these oxygen and nitrogen reactive species, which are molecules that are really good in killing the parasite. Okay, so but as I said, even with all these mechanisms, still the parasite has a way to avoid its total elimination. Okay, but we have to remember that every immune response is also come with some immunopathology. So when you're killing the parasite or the uninfected cells with the parasite inside, you also cause damage to the tissues, to the tissue around. So you have immunopathology, and this is very bad too. So the patient can die of immunopathology. So we need other mechanisms. So the inflammatory response and the cytotoxic response will has as a consequence immunopathology. And we need anti-inflammatory and regulatory responses to control the immunopathology. So it's really a balance that is needed. So in the last, so now I'm going to concentrate on the T cells, which are the subject of our studies. So CD4 T cells, T helper cells, came in different flavors. So after a naive cell is activated, recognizing the pathogen antigen, which is presented by the dendritic cell, then this naive T cell become activated and can differentiate into different kinds of T helper cells. We just talk about Th1 cells, which are the ones that produce gamma interferon, but each one of these subsets produce a different subset of cytokines with different functions. Th17 cells were also being seen in experimental models of T. cruzi infection in the mouse, but their role is still not completely understood. And also T regulatory cells, which are the ones that regulate the others and avoid autoimmunity. So T cells are also important. But again, a lot of, we need much more studies to really know what's everything that is happening. And so the question that we did is, can CD4 T cells also become cytotoxic like CD8 cells? Well, we don't find this in the textbooks normally, but yes, the answer is yes. Cytotoxic CD4 T cells have been known for a while, but they didn't get much attention until very recently. So as the title of this review article that came out last year says, the era of cytotoxic T cells has arrived, and T cells, cytotoxic CD4 T cells are present at the steady state in the gut, but they are also involved in different kinds of infection, in autoimmune disease, and in cancer. In our study that was published last year, we characterized for the first time the presence of cells, CD4 T cells with cytotoxic functions in the model, in an experimental model of Chagas infection with mouse infected with Trypanosoma cruzi. And I don't have time, of course, to go into the results, but what we found is that CD4 CTL, so cytotoxic CD4 T cells, were predominant among the cells that were infiltrating the heart of the infected mice. And it also showed that the adaptive transfer of T cells, with T cells are granzyme B positive. Granzyme B is one of the protein involved in the mechanisms of cytotoxicity, so it's a marker of cytotoxic cells, and perforin is also another important marker. And we show that these cells have increased the survival of susceptible mice. So maybe, but we didn't, we were not able to figure out which was really the mechanism behind this protection. Maybe, but we believe it is an anti-inflammatory role, because the cells also express a lot of immunoregulatory molecules. But importantly, we also look at patients in collaboration with Dr. Valderes Dutra from Minas Gerais, and we saw that the frequency of cytotoxic cells circulating in patients with the chronic symptomatic form of the disease, so the frequency of these cells was higher in those patients. And moreover, the frequency of these cytotoxic cells correlates with cardiomyopathy. And of course, we have to remember that correlation does not imply causation. So we have to understand more if these T cells are having an immunopathological role in the chronic phase, or if they are just enhanced because increasing in numbers because they are trying to control the immunopathology. If their role in the chronic phase is the same that we saw in the acute phase, meaning if they have an immunoregulatory role, maybe they are just enhanced because they are trying to control the situation, the inflammatory response. And we were happy to see that a few months after our study came out, another study from another Brazilian group in collaboration with the NIH laboratory also showed the enhanced frequency of these cells in mice, in patients with Chagas disease. And so the question remains: what is the role of these cells? What is their contribution? Do they kill infected cells, or they kill the presenting cells, which is another possibility? Well, so much work is still needed to answer this question. And coming back to the different subtypes. So a question is, are CTLs a new subset of T cells, or they derive from others? Because we also know that there is a lot of plasticity in the T cells, CD4 T cells, so meaning that one subset can turn into another subset, and this has been shown for several groups in several publications. So, and also there is evidence that CTLs may be terminal differentiated from Th1 cells, but we still don't know if this is always the case. Okay, so there's a lot of questions to be answered yet. Which is the specificity of these cells? Can they turn into memory, into tissue-resident memory cells? Can they be induced by vaccination? Which factors favor their differentiation? And importantly, to compare the cells in the acute and in the chronic phase. And also, I think a very interesting question is to know if these cells are a kind of regulatory cells. There is a subtype of cells that were called TR1s, so this question will be important for if we think that these cells can be used in a therapy. And for this, it's also important to know if we can regenerate these cells in vitro, and can they have a stable phenotype? These questions are very important if we think in T-cell transfer therapies. So I think maybe you've heard about engineered T-cells, which are used in in the therapy of cancer, mainly lymphomas. So these are engineered cells, but there are a lot of other clinical trials going on with the adaptive transfer of CD40 cells, T-regulatory or TR1 cells, not necessarily engineered, but there is a lot of hope that these therapies could improve the this disease, the treatment of this disease. And so the question is, can we imagine that maybe T cells can be used for a future therapy for chronic Chagas disease? So with this, I will end, and I just want to thank all the students, postdocs in my lab, and my collaborators in other universities in Brazil, and the agencies in Brazil that support that work. So thank you very much.